Okay guys, this is the lab about photosynthesis. What we are doing in this lab, we are separating the different type of chlorophylls from the spinach. Okay, I took some spinach and then I brought them, and then while you're grinding it, you add some 80% acetone right here. So you're adding, I'm going as I grind them, as I break down the cells of the cells of the spinach, I also am adding some. I added a lot this time. Sorry about that. Just a few drops, 1 or 2 drops, and then you keep grinding. Okay, so you after you grind and grind and grind, it's supposed to come, and then of course you broke the cells. I'm breaking the cells right now, and then I'm with the help of acetone, I'm separating the different type of chlorophylls. So when this is happening, as you're done like this, then after a few minutes of you grinding it, it becomes a soup like this. Okay, I hope you guys can see this. And then after adding some acetones to that, then I grab a, they call this a chromatography paper, and then what happens, I should not touch the other end, just like your pH paper, you're not supposed to touch it. So I hope there is one in here. Yeah, there are 2. Okay, 2. So grab one and cut it in a V shape. I hope you guys can see me, like that. You're not supposed to touch that end. And then from this soup of spinach and acetone, I hope you don't try this at you don't eat it. You grab a capillary tube. Again, I should be careful not to put this chromatography paper anywhere. You should grab a capillary tube from like this, and then put the lid on, and then you grab a drop, a micro drop, that is, you grab a micro drop, and then you put it in one spot right here. Okay, so that one was not a strong drop. Let's grab another drop, and right in the center, and then you dry it. And then while you're drying it, I'm drying it like this, so you dry it back and forth. You have to let it dry. If you don't let it dry, it will not work. And then another drop. Okay, now you can see it better. Let's drop right in the center, and then back and forth, back and forth, back and forth, back and forth. Another drop. So you have to do it 10 times. 10, 11, somewhere in that neighborhood. Another drop, and you and then after you've done this 10 times, 10 drops, again, I'm gonna put it here, then it's gonna become a chromatography paper like this, as such. I've done it 10 times, and then you put some 90% petroleum ether and 10% acetone. Okay, so I call this solution. You have to know what the solution is. The solution is ether acetone. And then grab another about 5-10 ml, somewhere in that neighborhood. I'm not measuring it right now. I should have, but I'm just grabbing an eyedropper and putting some in here. Okay. That's about 10 ml. And then put it in a test tube. I'm sorry, we are not like chemistry. We have explosions in here. We biologists are boring. In chemistry, if you had 3A, there I've heard. I have not watched those videos. And you put the stopper on top, as such, and then you put it in a test tube rack without shaking it too much. Okay, and it becomes like this. Okay, and then you put it in test tube back and you wait for 20 minutes or so. So at the end, when you wait it, it becomes something like this. Okay, so the different type of chlorophylls I have here. Okay, I'm done with this, and then everything has to be discarded. You have to go to biohazard bag and everything else. So on top, I don't know you guys can see this or not, on very top right here, that is xanthophyll. I'm sorry, on top is keratin. Right here, the light yellow, you can see that's chlorophyll, that is xanthophyll. Then this is chlorophyll A, and that's chlorophyll B. I hope I can point them in a way. So on top, right here, that would be, oh, sorry. Okay, right top, on top, that is your keratin, xanthophyll, all of that yellow, and this is chlorophyll A, and this is chlorophyll B. Okay, so you separated these, and you can cut these and put them in a spectrophotometer and find their wavelength. And here's another one. Oh, here is beautiful keratin on top. I hope you can see it, and xanthophyll, chlorophyll A, and chlorophyll B. All right.